All right. You actually want class on Tuesday?
Yes.
Amazing. All right. No. Congratulations, you made it this far. In this class, you're all still here. That's a good thing. Today we begin the last case study, which is about a Tarica granulosa, Thamnophis totalis predator-prey system in the Pacific Northwest. Did you have something to add, or are you just stretching?
Sorry, I'm stretching.
Okay, all right. You're standing, and
I know, I'm so excited about this actually. I haven't heard him say
this is actually my funnest thing that we do in this class. I designed this case study a long time ago, and it's been fun following how this particular case study has changed actually in the time that I've been teaching it, because the people who work in the system continue to do research, and so there's always something new coming up. Some of you have been working on predation as part of your lab projects, and so some of you are going to be more familiar with some of the material that we covered today and over the next 3 weeks than others of you are. Those of you who have been thinking about this because your lab project deals with predator-prey interactions, help out the people who have projects that don't deal with that. But before I get started on this whole case study, do you have any questions about lab? At this point this week, all of you guys have gone through basically Professor Nickel or myself picking apart your first drafts of your presentations. Was there anything unclear about what was suggested to you, or how to go about making those better, or anything like that? All right. Then we'll just begin. So.
Wait, there's one question.
Yeah. Oh, yeah. Sorry.
It's not really relevant to anything, kind of, but
Irrelevant questions, my favorite kind.
yeah. So, okay, we're learning up until the final exam. So the final exam, really get over this stuff and then start review, or is it completely cumulative, even out?
No, it's going to be just like taking a normal test. So we'll have 100 points basically on the stuff we're doing for the next 3 weeks. And then there will be 100 points that basically review everything that has come before. Or that's how we've done it. Dr. Reynolds and I haven't really talked about this in depth, but that's how it's been done traditionally. And I don't know.
Other times we've spoke about that, we've done a lot of the final session.
So no, it's a bear final. I mean, it's long, it's 2 hours. They block out 2 hours for it. So think of it as taking a normal 65-minute test and then taking another one. Yeah. Oh, not bad. I'm sorry, Chris is like, no, it's not bad.
No.
It's not bad. All right.
That's true.
Yeah. Okay. The person that I used to team teach this course with, when she taught, I would take notes about her teaching, and when I taught, she would take notes about my teaching. And she was really, really good about giving you learning goals at the beginning of every class. I am less good at that. Dr. Reynolds is probably somewhere between me and Dr. Schaefer. But basically, what we need to do today is we need to understand the predation sequence and predator adaptations and prey counter adaptations, and we'll get some feel for what adaptation is. You guys probably use the term adaptation fairly loosely, and then we're going to gain some insight into the process of science by reviewing the Brody chapter that was posted on iTunes U that you hopefully have read before you walked in here today. Interestingly, yesterday a reminder came up on Facebook. You know how memory came up on Facebook. You know how Facebook does these memories? Well, last year when we were getting ready for this case study, it turns out I had to be at a meeting during this time last year. And so Dr. Schaefer was teaching something that I would normally teach. And so about a week before that time, we sat down to go through what needed to be covered today. And because she has taken notes on my teaching in the past, she opened up her notebook that had notes from the previous year. And at the top of the page for this day's material, she wrote the topic. And then under goals, she wrote interactive story time. So here are some learning goals, but a third unspoken goal is this is interactive story time. Because what's the nature of the chapter that we asked you to read for today? You guys did read the chapter, right? Posted on iTunes, you? 2011 book chapter? What is that? Is that a primary scientific article? No. What is it instead?
Like a story.
It's kind of just a long story that's about science. Is it written for scientists? Does it read like it's written for scientists? Does it read like Veen et al. And Hendry et al. Read? No. Who's the target audience for that chapter?
Us. People who aren't. Like people who aren't scientists.
Well, are you putting yourself in that category, Claire?
No.
Are you in the process of becoming a scientist?
Scientist, Blair, come on.
Yes.
Yeah, so it's not really written for you. It's written for people not like you. People who aren't scientifically literate necessarily, or maybe they're scientifically literate, they're just not scientists who are formally trained. It's written for the general public, and so you'll see that the writing style is different. And the chapter has a much more story-like sort of feel to it. And the nice thing about the chapter, for a long time we had you read a primary literature article that was written in 1999 that kind of got you up to speed to what had been done in this system up until that point. And then when this book chapter came out, we thought, oh, well, that's a nice book chapter because it essentially covers the span of all of the primary literature that you'll read. So this is an unusual case study in that I'm going to talk some today about predator-prey interactions and the system in general. We're going to talk about regression analysis on Monday. After we come back from Thanksgiving, Dr. Reynolds is going to talk to you about quantitative genetics, and we're going to build on the quantitative genetics that you learned earlier in the semester. We're going to add another trait to it so that you can think about multivariate evolution effectively, hopefully. And then she'll talk to you about this concept called adaptive landscapes. And as soon as we're done with that, your first paper, which is Brody and Brody 1990, you will need to have a literature summary written of that paper. You'll bring that literature summary to class. We'll discuss Brody and Brody 1990, just like we have discussed other papers. And then the next day you're going to come in and you will have read Brody and Brody 1999. You don't have to write a literature summary about it, but you need to have read it and you need to be ready to come in and discuss it with your peers. And then the day after that you're going to come in and you will have read Gaffney et al. 2005. The next paper in this series of papers that tells us about this case study. And once again, you don't need to write a literature summary, but you need to come in ready to talk about the science that is in that paper. And then the last day of class you come in and you will have hopefully read Hanifin et al. 2008. And you'll come in and be ready to discuss that paper. So half of this case study is us teaching you, and half of this case study is you really teaching yourselves and one another, and it's all going to be done through primary literature. So you need to get geared up for after Thanksgiving. What are you going to be doing? You're going to be reading a lot of primary literature. Now, hopefully we've chosen good papers that kind of walk you through this process. One of the cool things about this system, and the reason that we teach it, is that it's about predator-prey coevolution, but it covers the topic all the way from large-scale evolutionary dynamics all the way down to the molecular mechanisms by which these two species are interacting. And so a lot of times when we think about ecology and evolution, we think of it as dealing with organisms and things like this, and then we think about cell molecular biology, and we think of that as the realm of cell molecular biologists. But the fact of the matter is you can study animal behavior and evolution, which is what this particular case study is about. You can study it at broad scale evolutionary scales, you can study it at the scale of individual interactions, but you can also study it on the basis of how individuals are interacting with one another and what the molecular biology of those interactions is like. And so that's one of the cool things about this case study that I think you'll find, I hope you'll find engaging at least. Bryce, do you have a question? No. Okay. Questions before we begin? All right, when I think about predator-prey interactions, this is a citation that comes to my mind constantly. This is a paper by Lima and Dill. The title of the paper is Behavioral Decisions Made Under the Risk of Predation. And this is the first paragraph of the paper. Just take a minute and read it for those of you who haven't already seen this paper. Everybody read it? When is the only time you should directly cite anybody, directly quote anybody when you're writing a paper? Repeat it louder. Actually, that's not what I think of citing people, quoting people
What do they
directly.
say?
Not believable or odd.
Well, I have told them that, so.
Oh, okay, so explain.
One of the times that you cite is when they say something that,
It's just so incredibly out there
It's
that
kinda
nobody
out
will
there.
believe it unless you actually quote it.
right, so the example that I used is during my dissertation, I included that Kurt Singer et al. Said that there's no species-specific lifespan for any species, like that there's actually not a limit currently.
And you found that so out there that you felt it necessary to
My
quote
advisor did
directly.
not believe me that they said.
Okay, all right. Okay, so that's one example. What's another example of when you should quote somebody directly? Yeah. Caleb. Well, why would you want to use somebody else's words exactly in the future? Isaac.
When it's such a simple idea that there's physically no better way of putting it.
When you have an idea and they've stated it in the most clever, pithy, rememberable way possible. And so these bolded words are words that I have repeated repeatedly over the course of my career whenever I'm talking about predator-prey interactions. So this paragraph is basically about everything that an organism is out there doing. One of the things that is constantly have to be in the minds of the animal doing those things is this overarching umbrella of the threat of predation. You're out there foraging. You have to think about who might be foraging on you. You're out there trying to get lucky. You have to be thinking about who might be out there trying to eat you, kill you, and eat you while you're trying to find a mate. Whatever you have to do, there's always this overarching umbrella of the threat of predation, the risk of predation. And few failures are as unforgiving as the failure to avoid a predator. Being killed greatly reduces future fitness. And it's short, and it's pithy, and it's clever, and I just like it. Yeah.
Basically, it's funny.
It's funny. It's funny. Being killed greatly reduces future fitness. Because if you're dead, you can't get lucky. All opportunities to get lucky are gone at that point. And so this is one of the groups who's working on predation. Actually, one of the members of the group found this paper, and I did not change this PowerPoint to include this quote. This quote was in here from many years past. So when you think about predation, when you think about what predators and prey do, you can think about it as a sequence of events. And this is from a book, a behavioral ecology book that I use in my animal behavior class. You can think about the things that a predator is doing, because the predator is the actor, the predator is the offensive individual in a predator-prey interaction. So predators search for prey, predators recognize prey, predators then go about the process of catching prey, and then predators go about the process of handling prey. And when we talk about handling, it's after you've caught something, but it's still alive, you still have to kill it and eat it. And sometimes you catch a prey item, and it manages to escape. Have any of you ever grabbed a rabbit suddenly or threatened the life of a rabbit?
Yeah.
Yeah, what does a rabbit do?
It's clean.
Oh my God, they let out this blood-curdling scream. I worked at a zoo when I was in graduate school, and we had reticulated pythons, these big 30-foot long constricting snakes, and we fed them full-size rabbits.
Oh my God.
Like not little bunnies, but like full-size house lap rabbits.
That is so sad.
Oh, it only gets sadder. Because the way you kill animals in a humane way is through cervical dislocation. You basically break their neck right. What's that?
I think kids in our school would break the necks of the rabbits and eat them if they weren't big enough to sell.
Oh my gosh. So the thing about rabbits is they have hardly any neck.
Oh yeah, the next one is tiny.
So getting a piece of wood over the neck of a rabbit and then grabbing its back legs and
Handsy piece of wood. You just break with your hands.
They're big. These are like 30-pound rabbits.
Rabbits were what I did was great for our main source of meat. It was my job when I was 8 to butcher the rabbits.
Well, I wish you had been with me when I was at the zoo. So the first time I go to cervically dislocate a rabbit, a full-grown adult rabbit, I pull up on the legs, and this thing lets out a blood-curdling scream that startles me to the point that I make the mistake of letting go of the back legs. And as soon as I do that, the rabbit is off. So there's this enclosure behind the reptile house at the zoo that I worked in, and I'm chasing, there's no way to get out, it's totally fenced in because everything in the zoo is fenced. And I'm totally running around the yard, chasing this rabbit around the yard, and then the zoo director comes around the corner and he's like, "What on earth are you doing?" I'm like, "I'm trying to feed the reticulated pythons." He's like, "Rabbit got away from you, didn't it?" I'm like, "Yeah." So they let out this blood-curdling scream that basically allowed that rabbit the split second it needed to escape me. I already had it caught. I was on the verge of killing it, and then not eating it, but feeding it to somebody else. And it let out this huge scream, and it made me let go. That's the difference between catching prey and actually handling it, killing and eating it. So what I want you to do is take a couple of minutes in your groups and figure out what are the kinds of predator adaptations that help a predator search for prey, recognize prey, catch prey, handle prey, and what are the kinds of counter adaptations that prey might have to counter the adaptations of predators. So go through that and essentially get off the rest of this table. Do it. I don't know that I could get my hands around these rabbits. They were so large.
They're really big, but it's much harder when they're that big. And the next are really tiny, so we usually there's no eyes. And so then you talk about snakes. And you're like,
So just do your task. Fill out the table. So chemosensory. So keen sense of smell.
Oh, yeah,
okay, so adaptations.
Yes, what are the adaptations and counter adaptations?
What would they have if that like means they can't be found?
Just like from them.
But would that be recognition or search?
Access the environment.
To do it that way, because it might not give the new dislocation.
Well, like couldn't they both have the same? Yeah. Or if it's like try to stay, avoid predation by doing the same things the predator does.
We had a new site into that.
So it's probably. I thought you were going to say that live.
Oh, no.
Animal care.
No, you don't ever see reptiles like that. On the search space, probably not.
Predators that can't move fast enough to catch that don't necessarily be this particular strategy.
Or not maybe not better strategies, but other
Doesn't
strategies.
have any natural predators because a lot higher in the
Are you guys talking over me?
Initiative Prey.
Go.
We'll catch it for prey, that would be stuff like, yeah, Callan.
Yeah, so in some cases, the adaptations interact with incredible visual
Weird snapjaws.
insight.
The lockjaw. Like hyenas have those too. So like their prey can't get away, but they don't kill it. I know sometimes I feel like sometimes like when they catch and handle it, they catch it and handle it like big cats grab the neck usually. I mean they can do it other ways, but that's usually what they do. Counter adaptation would be like they have like scales or like
No fair looking
spines
at
and
that.
stuff.
It's not fair if you've downloaded the PowerPoint or looking at the slides ahead. That's like kind of cheating, because all the answers are there. It does, as a matter of fact.
I would say it's a lockjaw. I don't really know. I mean, if you poke, like, if you poke, like, them in the eyes, I know they, like startles them. But I don't know how, like, an animal would do that.
I mean, like, dogs are like poisonous, right? Some have to be like poisonous.
Oh, that's true.
Tasting sugar. That's how poison stuff.
Yeah.
Is they eat certain like fermentation.
I don't know. It's just like hard because like, well, it's like something like a shark is gonna eat you. You're probably not gonna get away.
Swim backwards.
Unless they were bigger, yeah.
You can have a new version.
Yeah. Is that
Okay, what kind of things you kill things in large jaws? Okay, so those kind of offensive sort of weapons.
What else was that like the hand of prey?
So they mentioned things like, powerful jaws, offensive sort of weapons, but you can also think about what are the things that might enable you to escape as a prey item, and do you have the ability to evolve adaptations that counter the things that prey do? All right. Given some of what I've heard, I think maybe it's time to move on. All right. So the first thing, searching for prey, the kinds of things that help you search for prey is things like visual acuity. A search image. You guys know what a search image is? A search image is a kind of a mental image that you carry around with you that tells you, that informs you what the thing you're looking for looks like. If you came from a culture where people don't have cars and don't have locks, and you misplaced your keys in your house, and you said to this person from another culture where keys don't exist, I've lost my keys, help me find them, would that person be very well equipped to find your keys? No, because they don't know what a key looks like. You're better equipped because you live with keys all the time. And so a search image is this visual image, this mental image that you have that tells you what something looks like in your environment. Searching where prey are abundant. Counter adaptations by prey are things like crypsis, Lots of different kinds of crypsis. Polymorphism, looking like something else or looking different. And sorry, polymorphism isn't looking like something else. Polymorphism is looking different enough within your population that it kind of confuses a predator because he never knows what he's looking at. Spacing yourselves out, becoming less dense. Have you guys seen the movie Shooter?
Yes.
Mark Wahlberg, great actor of our time. That's Sart's Alex. Shooter, towards the end of the movie, Mark Wahlberg is in the snow. He's basically going to kill people who are down there. He is here on the white snow, and he's wearing this thing called a ghillie suit. This is what a ghillie suit looks like when you're not up against white coloration. And basically, this is what we call disruptive patterning, disruptive coloration. It's basically breaking up the outline of his body so that he doesn't look like a human being. He just looks like a lump of snow. This is one form of crypsis. Once you've found something, you have to recognize whether or not it's prey, whether it's not it's safe to eat, whether or not you want to eat it. And one of the best things that a predator can do is learn about the potential prey items that are in its environment. But you can masquerade, if you're a prey item, you can masquerade as something that is not edible. You can have warning coloration, aposematic coloration, that signals to the predator that you might be dangerous or toxic. Mimicry is involved in this because you can mimic something that looks like something that is harmful when you yourself are harmless. A classic example of aposematic coloration is coral snakes. Coral snakes are oftentimes banded with red and black and or yellow banding colors. This is all from a textbook, or not a textbook, but just a book about venomous reptiles of the Western Hemisphere. And these are all coral snake patterns. So you might have learned, I don't know, sometime in your life, what's the rhyme for knowing how to recognize a coral snake? Red on yellow, kill a fellow, red on black, friend of Jack. Guess what? Don't go to Central America and use that, because you will pick up a black and white banded snake. You will hold it up over your head to your colleague who has asked you to go collect snakes with him, and you'll be like, I've got a non-venomous snake, I've got a non-venomous snake, and the other guy is yelling, put that down, you're about to get bit, and it's going to be ugly. So these are all coral snakes. I'm going to repeat this page here and put another page up. That first page is there. These are all milk snakes in the genus Lamprocultus for the most part, and these are all mimics. So you can have warning coloration because you're dangerous, but the other thing that you can do is you can mimic dangerous snakes so that when you're in Honduras, you can pick up a Honduran milk snake, which is harmless, or you might make the mistake of picking up whatever this snake is, which is the Honduran version of a coral snake. And so mimicry can also be one of these situations where you basically either warn an individual that you are dangerous or mimic the warning coloration of somebody who is dangerous. Catching prey. You can sneak up on the prey. You can be fast and be able to track them down. You can have weapons of offense. You can have claws that you use to grab them and things like this. One of the interesting things is you can just signal to a predator as a prey item that basically says, I see you. I see you over there. And you're so far away from me. Now that I can see you, I can get away before you can get to me, even with all of your speed. Escape flight. The startle response like rabbits do. Deflecting attacks. You can have weapons of defense. The most interesting to me is the signaling to the predator that it's been detected. If you've ever seen white-tailed deer run away from you, they put up their tail, which is white underneath. That's why they're called white-tailed deer, which is weird because it just makes them more obvious. But what that is it's a signal to whatever it's perceiving as a predator that I've seen you and I'm running away from you, so don't even waste your time. Gazelles in Africa do the same thing. It's a very stereotyped behavior called stotting. They jump on very rigid legs up and down. They're moving forward slowly, but they're moving forward really slowly. But they only do this when they have located a predator in their environment, and it's their signal to the predator, I've seen you, don't even waste your time. Because if you start coming towards me, I'm going to stop stotting and I'm going to start running, and you're never going to catch me. Isaac.
Is this similar, is then the rattling of a rattlesnake like a combination of this and also a warning?
It's mainly a warning. It's a form of auditory apposematism. And then when it comes to handling is sometimes the most difficult for students to think about because it's your ability to subdue and kill the thing. And so you can have things like active defense, you can have the spines of horned lizards and things like that that come into effect to keep you, after you've been caught, from becoming dinner. The one that we're going to talk about in this class is toxins, because we're dealing with a newt that produces a toxin. And one of the things that can evolve in predators is the ability to detoxify whatever that protective toxin is of the prey. So whenever you think about predation, you can think about it as a sequence of events. And at each step in the sequence, predators can have adaptations that increase their probability of getting food, and prey can have adaptations that increase their probability of not becoming somebody else's dinner. And we expect selection to be higher on prey than it is on predators. Why? Why would we expect selection pressure to be higher on prey than it is on predators? Somebody other than Isaac. Right.
Because they're adapting to both the environment and also being killed by the predators.
And what do we know about being killed?
It stops.
Decreases fitness.
It greatly reduces future fitness, doesn't it? And so prey are under greater selection pressure than prey, or sorry, than predators. This is summed up in what's come to be known as the life dinner principle. Selection is higher on prey because if a prey individual loses the predator-prey interaction, it loses its life. If the predator loses the interaction, what does it lose? Food. It just loses its dinner. And so for this reason, the life dinner principle, you would expect selection to be higher on prey than it is on predators. This becomes crucially important in this case study because in this case study, it's a case study where the life dinner principle doesn't apply. And so you can have equal amounts of selection on predator and prey. And we'll talk about that in a little while. So this is the dude that we'll be studying, one of the two species that we'll be studying. This is Pterica granulosa. This specimen is from Oregon, collected by me last year. What do you notice about this guy other than that he's adorably cute? Yeah, again. Okay, so he's darker on top than he is on the bottom. We'll see what his underside looks like here in a few minutes. That serves as a form of what? That dorsal coloration. Crypsis, because when he hasn't been artificially placed on a bed of moss for taking a staged photograph, a lot of times they're walking around on the forest floor and there's a lot of dirt and stuff on the forest floor or dead leaves, and so they blend in pretty well. What else do you notice about this guy? Speak up. He doesn't have claws because he's an amphibian, but he does have little pointy feet. Claire, is your hand up? He does have a really long tail. When they go to breed, they are in water and they swim and they use that tail for locomotion in water. What else? Isaac.
He seems kind of moist.
He isn't all that moist. He's moist on the bottom here, but he's actually kind of on the dry side for an amphibian in reality. Asa.
And these are like web.
What's that?
That's a web.
Web? Or wet?
Web.
Webbed. I don't think he has webbed feet. He doesn't have webbed feet. What's that? Gabe. He has bumps all over him. What are those bumps? Pores from what? Well, the bumps aren't the pores, but the bumps are the things that are the pores are facilitating. They're secreting something. What are they secreting? Toxins, because this is a toxic kind of newt that has toxins as its defense mechanism. You should have learned this from your book chapter. Tarica granulosa. What does granulosa imply? The granulated newt, the bumpy warty newt. You guys ever? What is the recipe for every witch's brew? Eye of newt. Newts are well known. Members of this family, the family Salamandridae in the amphibians, are well known for being toxic, hallucinogenic, behavior altering, or deadly, depending on what you're thinking of. These guys exhibit a very peculiar behavior when they get attacked. They go through a thing called the Unken reflex. The Unken reflex is basically something amphibians do, where they toss their head back, they toss their tail up, they generally put their legs out, and what it reveals is it reveals this bright coloration on the underside, so that if you're a bird and you try and eat a newt and it makes this display, you have this bright color in your head, so that in a couple of minutes when you get paralyzed and fall over, you remember that bright color, and the hopes are that you won't come back and try and eat one of those newts in the future. And it's a very stereotyped response. Do this on 50 newts, and if you pinch them hard enough, they'll do this. Every one of them will do it, and they'll do it all in pretty much exactly the same way. This is Edie Brody III, Little Butch, people who know him. His father is Big Butch. They're both large men, so Big Butch was the Big Butch the elder, because he is Edie Brody Jr. This is Edie Brody III. This is him in Oregon, once again, collecting snakes. This is the garter snake that he worked out for his doctoral research. Big Butch was on my doctoral dissertation committee, and so that's one of the reasons that I know about this particular case study. This is his son. When I was in graduate school, he was just in the process of finishing up his PhD work at the University of Chicago, and then he went to the University of California at Berkeley and did a Miller postdoc for a couple of years. Then he taught at the University of Indiana, and now he is at University of Virginia. So on iTunes U, there are a series of discussion questions. There aren't very many of them. Pull those up. Spend a couple of minutes going through those. They're all questions that have to do with the chapter that you were supposed to have read for today. Dr. Mills, how rigid is your definition of adaptation? An adaptation, what an adaptation is as a trait, as a character.
I think it's pretty hard to show something that is adaptive.
Yeah.
So you have to
Please do.
So who was wrote back in the early 80s? Adaptation, if this applies. Undertaking adaptation. These
So
are
what is he doing? How many years that's what snakes were important to
Yeah, I have read it because
progress we did for them.
That's what we did.
I know this was like, they were talking about, well, they were eating news, or like they would like eat them and like hold them in their mouth and like spit them out. And they were like measuring, and that was like, this was, they were like measuring. Because he was like, what the name is? So yeah, he was like, so there was like, I can't remember what they were doing, but I was like, I just was like physically like making them puke. Okay. So they were, yeah, they were like, yeah, so like, they would eat like, they were eating the salad, and then they would like barf, and then like they were like studying the stomach contents of the snakes, and then I saw that like some of the like toxins which were like in their stomach. I was like, oh. But yeah, he was like to study for snakes, to study the like stomach contents and like measure the
Rose, 2 of the Vian et al. Peer reviews got in with a stack of Vian et al. Papers. I need to give those to you.
Gotcha. I have this one for you
Yeah, they were like, they were barking. What's the
What was funny?
When they were barking the snakes.
Oh, yeah.
Yeah.
It turns out you can do a lot of things with the snake because it's just basically a
Yeah.
long tube. So you can actually force it to regurgitate things that it's already eaten. One of the other things you can do is as you run your thumb down the belly, if it's a gravid female, you can count the number of offspring she's forming because they give live birth, and so
Yeah.
their embryos are growing inside. And actually, on this picture, you can see little lumps inside of the snake. And so you can palp the snake and actually count the number of offspring that she's going to have in the future.
How many do they have?
I don't know. I would have to look it up.
I think some people are talking about his dad's. Well, yeah, he's talking about his dad's research, but also with the like the campers who died, and the only thing they found was like a newt in their like and so like they had like dirty toxins and like died. It's like really important actually. They did. Yeah, I don't know either. I don't like snakes.
I was fine with them until a couple of days.
No, those guys should stand.
And it was a little bit of an artery.
I don't like anything that crawls.
Oh, sorry.
I just did not provide them. In this shop.
And I was feeding them to and she was chilling. And then I had a couple of them. So I started walking once in a while. Wanted to make sure to that's so it bit me and then it realized what it was. Because he was
It's like they can yeah, it's like they can get this and stuff they don't. Like they recognize it like
she's still there. Yeah.
What are some of the salient characteristics?
Because it missed the marks, the pinky, and it would not let me go because it would see the pinky.
What is-
So you need to relax.
Characteristics kind of.
Get him.
His dad studied the toxicities and like trying to match the resistance of like to the news. So like, it's like later he like hooks up with his dad and they do like a mutual. But his dad, what do you think say about because I know that he sent the like he bred those little like baby snakes.
His father didn't, he did.
Yeah, he did, and then he like sent them to his dad. But was his dad just doing the resistance before?
No, what his dad did when he was an undergraduate student back in the 1960s was he fed a bunch of newt skin fragments to all kinds of different animals to see if they died.
That's so sad. I feel like that's not human.
It was a fairly crude paper because he fed it to a lot of different kinds of animals.
Yeah.
And in some cases it took them, an hour to die, but in some cases it took them days to die. In some cases they didn't die at all. And he did a wide range of different kinds of animals. It's the kind of research that you could get away with in the 1960s.
Yeah.
You couldn't get away with nowadays.
Like trapping people in prison since I'm getting an LSD.
All right, so it sounds like things are settling down.
It's just the animal.
So when Little Butch was doing this research, when he struck upon this idea of snakes and the newts, what was he actually doing? He wasn't studying this to begin with. He was actually studying something else. Yeah. Sorry, I didn't see.
Collecting reptiles with mostly garter snakes and studying the pattern of natural selection.
Yeah, so he was actually studying. You'll notice in this picture that most of these snakes are striped, but in the same populations there are snakes that are checkered. And he was interested in why these two color patterns would exist in the same species, in the same population. And what he found, and Dr. Reynolds might or might not talk about this later, is he found that there was different levels of selection on color pattern and behavior. So striped individuals that reverse themselves got killed a lot out in nature. Checkered individuals that ran in a straight line got killed a lot. But if you were striped and ran in a straight line, or checkered and reversed yourselves a lot, you tended to have higher fitness. So he was studying something totally different, but as he was out collecting these snakes, what kept happening? He would occasionally find what? He would find either dead or immobilized snakes. And he would take them back and he would cut them open to try and get an idea of why they were dead, and what would he find? He would find newts in their guts. One time, he put what he thought was a dead snake in his bag, and when he got back to his camp, what did he find in the bag? No, he found a dead snake. What else did he find in the bag? He found a newt that had crawled out of the dead snake's mouth because the snake had swallowed it. And then he picked up the snake, put it in his bag to take it back to his camp and dissect it. And in the trip, the newt had crawled its way out of the snake's mouth and survived the predation attempt by essentially killing the snake. And so after this happens enough, what does he remember? Well, he knew newts were poisonous because he grew up in a family of herpetologists. He remembered something specific, though.
Oh, do you mean he, Brody, or he, the snake?
He, Brody Brody the Third. He, Brody the Third, remembered something as he was making these observations. What did he remember? He remembered the story that his dad told him when he was a kid about when Butch Brody Jr. was an undergraduate. It's one of the first examples I know of actual undergraduate research. There are other examples. Who's the guy who did linkage? Why can I not remember the guy's name? Geneticist. Linkage mapping. Sturtevant. He worked on linkage mapping as an undergraduate, strangely.
Yeah, in Morgan's lab, yeah.
In Morgan's lab, yeah. So there was a time when undergraduate research wasn't really much of a thing. This was one of those times. And the dad had been at Western Washington, and there was a guy there named Bob Storm, and Big Butch, Butch Jr., walked into Bob Storm's office and said, I would like to get involved in research, I would like to go to graduate school. Do you have anything that I could work on? And what did his professor tell him about? Yeah, told him about the incident of the coffee pot. Were they hunters or fishermen? I'm trying to remember now.
Yeah, all I had heard, I think.
Well, it tells you in the story. Well, anyway, hunters or fishermen, they were camped next to a stream. What happened? They went down to the stream, scooped up some water in their coffee pot, threw the coffee in there, set it on the fire, drank the coffee, and at some later time, somebody found the 3 people dead. And when the cops went to investigate, the only thing that they could figure out was that when they looked inside the coffee pot, there was a newt that had been boiled alive in the coffee pot, and so they attributed it to accidental poisoning by this newt, but they didn't have any evidence. They didn't have any, it wasn't like CSI where you can do a tox screen on the individual and figure out what they actually died of. And so the newt was implicated, but they didn't actually, that was an assumption more than anything else because there was no other signs of foul play. And so Bob Storm basically said, I read this newspaper article about these 3 guys that died next to a stream. The only thing that was implicated in the deaths was a newt in the coffee pot. Figure out if that's what killed them. And Big Butch went off and tried to do that. How did he go about doing it?
Killed a bunch of little animals.
In 1968, he published a paper where he basically took skin off of a bunch of newts, fed little flaps of skin to a wide variety of organisms, and then waited to see how long they died. Recorded their weight, recorded the weight of the skin that he fed them so that he could get some idea of dosage, and basically saw if these newts were capable of killing or immobilizing a wide variety of vertebrates. He used domestic cats, he used rabbits, he used mice and rats, he used natural animals from out in nature that he found. One of the things that he also tested were snakes that he knew from growing up in Oregon were known to prey on these newts. And what he found was that some of the species of snakes that he tested didn't die at all. And then he published that paper and went off to graduate school at Clemson, and then has studied predator-prey interactions and chemical defenses ever since. But he kind of stepped away from this because he was all the way across the country and the newts only occur over on the West Coast. So this is Big Butch on the left, Little Butch on the right. Little Butch's, I think that's his oldest son now, but he's much older than that now. If there's any mistake that genes affect phenotype, this should remove those doubts from your mind. These guys have what we refer to as the Brody-Campbell morphotype, because in my graduate program there was Butch Brody, Big Butch, and there was this guy named Jonathan Campbell, and whenever you saw them at a distance you could not tell them apart because they both kind of looked like that. Very large men with bushy beards and graying hair.
They're a subset of the ecologist stereotype that we spoke of last night.
Yes, yeah. So a friend of Big Butch's named Roy McDermott, who is at the Smithsonian, they all have the Brody-Campbell morphotype. And when I first saw Roy McDermott at a meeting, I'm like, oh, we should expand it. It's the Brody-Campbell-McDermott morphotype, and it is one of the common subtypes of ecologists. Anyway, this is them in Oregon, near places where newts hang out and actually catching newts with dip nets, which is a very effective way of catching newts. So we talked about what his father did and what his father found. His father found that these newts kill a great many kinds of animals if you feed the skin of the newt to the animal, but snakes, at least certain species of snakes from certain areas, have an amazing resistance to this toxin. And so little Butch was in the graduate program at the University of Chicago studying quantitative genetics, and when he kept seeing these snakes barf up these newts or finding snakes that had been immobilized by newts, et cetera, he got interested in this thing that his father had done back in 1968, and they started teaming up on this project to try and get at what is the evolutionary interaction between a toxic predator, a toxic prey item, and a predator that has seemed, at least in some cases, to have evolved resistance to the toxin of the prey. And so this is a fairly famous photograph of Little Butch's, essentially showing a snake rolling the dice, playing Russian newt roulette by engulfing a pretty good-sized Tarica granulosa. And as time went on, Little Butch and his father, Big Butch, did a lot of the work themselves because it was all kind of behavioral work that they were equipped to do. Butch Brodie Jr. is basically a behavioral ecologist who studies predator-prey interactions. Little Butch is an evolutionary biologist. His specialty is quantitative genetics and studying life history characteristics, as well as also predator-prey interactions. But neither of these guys are molecular biologists, and they took their research as far as they could go with the tools that they had available to them and the skill set that they had available to them, which was basically injecting snakes with tetrodotoxin at known quantities. And then what did they do with them after they injected the snakes with tetrodotoxin? I do this with one of my undergraduate research students. Race them down a racetrack, see how fast they go. And what happens when you inject a snake with tetrodotoxin? It slows it down. Even if you're resistant to the toxin, the tetrodotoxin still slows you down. Remember that tetrodotoxin is a nerve blocker. It blocks sodium channels in nerves. And so when you block sodium channels, your nerves aren't firing as efficiently, or they're not firing at all. And so that then slows down the signals that you need to make your muscles move in a coordinated fashion. And for a snake that is slithering, the musculature of a snake is really complicated and requires a lot of neural coordination. And so they would race them down the racetrack, found that some snakes were not immobilized very much at all, found that other snakes were totally immobilized, and they saw everything in between. But that only gets you so far, and they started to have questions that required them to do what?
See if it was like a neurobiological issue or something that was going on in their brains.
Yeah, they needed to start looking at what the actual functionality of this system was. What exactly is the tetrodotoxin doing? What is it affecting? At this time, we already knew that it was a neurotoxin that blocked sodium channels, but they didn't have any idea whether or not the snakes that were resistant had sodium channels that were resistant, or if it was something else that was leading to resistance. Maybe it was the fact that they just didn't absorb it through the lining of their gut or whatever. So they didn't know what the mechanisms were, and so what did they have to do? Did they just say, I don't know about that stuff, I'm done? You guys back there know, because we talked about it. They collaborated. As soon as they got up to the point where their knowledge and skill set couldn't take them any further, they didn't just give up. They went and found people who knew the kinds of things that they didn't know but needed to know in order to ask these additional questions. They went and got together with people who knew about neurophysiology and knew about how to get genes out of one organism into another organism so that you can then control what the phenotype of that organism is so that you can test it. And so they took snake genes and took them out of snakes and put them into frog eggs and then tested to see if those frog eggs were effectively blocked by tetrodotoxin. If those frog eggs, sodium channels rather, were blocked by tetrodotoxin. They collaborated with Fred Jansen at Iowa State, University of Iowa. Ames. Iowa State. He's at Iowa State. He's a phylogeneticist, among other things. They needed to know what the evolutionary relationships of these snakes and newts were. So they collaborated with him so that they could get that evolutionary information to see how these things are evolving over time. Whenever they butted up against an area where it really wasn't their field of expertise, they went and found people who had that expertise and recruited them and said, hey, we've got this cool project. Would you be willing to come and work with us on it? This idea of collaboration is kind of the norm now in science. It wasn't the norm in science in the 50s, 60s, and 70s, but it really is the norm in science today. And so one of the characteristics of scientists is you have to be flexible and you have to be collaborative because it's impossible for you to know everything. And sometimes the questions that you want to ask and the most interesting questions that you have take you outside of what you're classically trained to deal with. And so this story is also useful to read, not just to get you ready for the next 3 weeks, but also to give you some insight into how science works. At each step along the way, when they got an answer, did they just stop? Yeah. Every time they got an answer to the questions that they had, that opened up a new set of questions. When we talked about the scientific method back at the beginning of the semester, what were the steps? Observation. Question generation. Do you have questions about those observations? You have a hypothesis. You have a set of proposed answers about those questions.
Experimental design.
You designed some experiments that get at those questions. What else do you do? Do the experiments. Next. Analyze the data. Last. Draw some conclusions from that. Is that the last thing?
Condense, you have to critique.
The conclusions from one study are your observations for your next study, and then you just delve further and further and further into the thing that you're studying. And over the next 3 weeks, we're going to go down that path in this particular case study. Questions? See you on Monday.